The third subtopic is application of high throughput technology in fermentation. This is another concept that has been introduced in bioprocess and in fermentation. So what this technology is all about, it is a technology that allows the conduction of parallel experiments. So that's what the term high throughput implies. So it means that it involves the conduction of many experiments, or in the case of fermentation, it deals with the bioreactors at the same time. Yeah, so that's why high throughput is coined the term. So the throughput actually refers to the quantity of the unit operations. So the number of parallel experiments depend on various formats, brand of reactors, and also the need of users or the applications, whether it's for microbial fermentation, for animal cell cultures, or for plant cell cultures. The common formats are 4, 6, 12, 24, 48, or more. Yeah, the number can be higher for automated system. So this number is referring to the number of, in the case of fermentation, the number of bioreactors, or the number of wells that can contain the ration. Perhaps it's not just fermentation, but the platform can also be used for enzymatic reactions or biocatalysis. So this technology is initially introduced for analytical purposes. Before it has been introduced to fermentation, it is used for analytical purposes. So what is meant by analytical purposes is for the analysis of, for example, proteins, carbohydrates, or glucose, or other metabolites. So since the analysis normally involve high number of samples, so the high throughput platform will facilitate the analysis procedure, because a lot of samples can be analyzed at the same time. But throughout the time, the technology, the high throughput technology, is also introduced in other parts of bioprocess, including fermentation. Yeah. All system like a single bioreactor is referred to as low throughput system. Yeah, because it exists in only a single unit. Yeah? For example, if you see the picture at the bottom here, this is a single bioreactor. So it is considered as a low throughput system. The second picture here shows high throughput microbioreactors, which exist in 24 parallel individual bioreactors. So this system is considered as high throughput system. Okay, so that's the difference between high throughput and low throughput system. In a comprehensive system, the high throughput system is connected to a computer that communicates with the embedded system to acquire data and set up experimental variables. So because the number of the unit operations is high, so it will be better if, let's say, the system is connected to a computer, which is always the case because these modern bioreactors, of course, they have the compatibility with a computer. So normally they are connected to a computer, and the monitoring, the control can be done from the computer. And even it can also be controlled or monitored remotely by using mobile phones or mobile devices. In facilitating early stage process development of any new products, the exploitation of HTTP models is of great interest. So this platform, high throughput microbioreactors, if we talk about fermentation, they are designed to support early stage process development. So what is meant by early stage process development? It involves preliminary studies such as screening and optimization studies for the development of any new products. For example, before any vaccines, like for example vaccines or any other types of products, before they are produced at large scale, they have to undergo certain stages whereby those, for example, like the conditions, those culture conditions need to be identified. Which temperature gives the best product title, which pH, which media, which microorganism, and so on. So this stage is called screening and optimization stage. So this is actually the early stage process development. So it doesn't actually require mass production of the end product yet. So that's why high throughput microscale bioreactor is preferred over large scale bioreactors. So the benefit of using high throughput microbioreactor is that it will promote rapid data acquisition, and in general, it will shorten the process development timelines. For example, if you see this platform, it has 24 microbioreactors. It means that 24 fermentations can be conducted at the same time. So instead of like running one by one using low throughput system, like 24 fermentations, 24 times of fermentation, so it will take some time actually for us to use a low throughput system compared to the high throughput system, where we can run 24 fermentations at the same time if we use the high throughput system. So the first picture here shows a high throughput system that is connected to the computer. And if you see these white things, white stuff on top of the, this one is actually the whole thing here is the laminar flow, whereby it will provide sterile environment. And inside this laminar flow are all those high throughput microbioreactors. Yeah. So this is another example of a high throughput microbioreactor system. So there are actually a lot of models, brands, designs that are produced by different companies or manufacturers because this technology, similar to microscale and disposable technology, it has been around for some time already, for at least like 20 years. So throughout that 20 years, there are many brands, many models of bioreactors that have been produced. So these are some examples. It's not the only type of high throughput microbioreactor. And I forgot to mention that high throughput technology, disposable technology, and microscale technology, they are normally integrated together in a single platform. So that's why you can see, like, for example, Micromatrix system here, it has 24 individual bioreactors, which means that it has the high throughput feature. And apart from that, the volume of each reactor here is less than 10 milliliter, which means that it has the mini scale feature. And the material of each of the bioreactor is made from disposable material. So it means that it is only to be used for single time, single use. So it has all the three phases, microscale or mini scale, disposable or a single use, and also high throughput feature. Okay, right. Application of high throughput technology in fermentation starts with the use of conventional microwell plates. So if you see the picture on the right here, this is the oldest type of high throughput platform, and this platform is called microwell plates. Yeah, and you can see that there are different number of wells inside this platform. Yeah, so there are many wells. For example, here the wells are much bigger than the rest, and there are 6 wells in this one platform, right? In this one microwell plate, and for this one there are 12 wells, and each of that column is called wells because it can contain the reaction or the fermentation. So this one is much smaller. I think this one is about 1, 2, 3, it's 24. And this one, I guess, is maybe more than that. 24 or 48. Yeah. So there are different sizes, different number of plates, or no, different number of wells, yeah? Depending on the size or the volume of each of the wells. Right. So this conventional microwell plates, it doesn't have the control system. It doesn't have the pH control system or the temperature control system by itself. So in order to use this, it has to be placed in this equipment called Microwell Incubator, where this equipment can be closed, yeah, and it can provide ambient temperature for the plates that are placed inside it. Yeah? So these are the plates, the microwell plates placed inside this Microwell Incubator. So this incubator, it can also provide shaking. So in order to mix the reaction mixture inside this microwell, yeah, but it doesn't have the pH control system and also the dissolved oxygen control system for the aerobic reaction, for the aerobic fermentation. So because of that limitation, people have improved throughout the past decades. They have improved this platform by inventing the microbioreactors. The one that I show here, these are actually more modern than the conventional microwell plates just now here. Yeah. So throughout that 20 years, there are many actually improvement that have been made. Yeah? And the use of high throughput technology have been reported in the literature. And these are some of the works that reported the use of high throughput technology for various applications, not just for fermentation, but also for biocatalysis. Biocatalysis is the enzymatic reaction, and also for downstream processing. Yeah. So fermentation is upstream processing. Downstream processing is something related to separation and purification of the product. Yeah. Right. So this is the conventional platform, and towards the recent technology, people have invented control microbioreactors. Okay, I think I've mentioned about this. And control microbioreactors are much better than the conventional microwell plates because they have the individual pH, temperature, and dissolved oxygen control system compared to these conventional microwell plates. But these microwell plates are still considered as high throughput platform because it has more than one. It has 6, 12, 24, and yeah, and so on. Okay, here are some examples of commercialized microscale bioreactors. So this is Micro24, the name of the microbioreactor, and it is produced by PAL Corporation. And this bioreactor is not really the latest edition of microbioreactor. It has been invented back in 2007, if not mistaken. So it has been around for some time, more than 10 years. Yeah? And this Micro24, how to operate this, it has this cassette. If you see this stuff here, this is called cassette, and this is actually the detail of the cassette, whereby it has 24 wells. Yeah? So this one is not in sterile condition because this is just for the for the photography purposes, yeah, just to show you how the wells looks like. But it has to be, it's supposedly to be handled under the laminar flow for the sterile condition, if let's say it is to be prepared for the fermentation. So underneath this cassette, the whole unit here is called cassette. Yeah? So the wells are actually integrated together under this one cassette. So underneath this cassette, you can see that there are dots. There are dots, white and slightly red dots. So those dots are actually pH sensor and temperature sensor. So that is how the pH and the temperature can be controlled individually in each of these plates, in each of these microwell. Each of these is called microwell. So there are 24 microwells, and it has to be covered with this lid, which has sterilizing filter, the one that is white in color here. So that filter allows the passage of oxygen and carbon dioxide. So for example, if let's say it is for aerobic fermentation, then oxygen will go through the filter. Yeah, so it means that the oxygen will be filtered. Yeah, it will be sterile once it reach the interior side of the microwell. Okay, this is the overall setup of the Micro24 reactor. So you can see the whole system, the same equipment here, which is next to the computer. So the computer is used to control the condition inside the microwell, yeah, the pH, the temperature, and also the dissolved oxygen. And these are all the gas cylinders which are connected to the equipment over here where the cassettes are located. And the ammonia generator here is also connected to the Micro24. This one is used to control the pH, yeah, by sparging the ammonia. That is how the pH can be controlled inside the microwell plates. So this one, it has the pH sensor. So this thing, when it is connected with, when it is placed on this platform, it has a certain channel that connects to the ammonia. So that's how the ammonia can be channeled inside this microwell plates. So you can see that this equipment is actually not that big. It's not as big as a conventional conventional bioreactor. It is small in size, but yet we can use the whole system here to carry out 24 fermentations simultaneously. Yeah, so that's the great side of this high throughput microbioreactor. Some other examples, these are some other models produced by other manufacturers. This one is Microbioreactor Amber by Tap Biosystem. So this one is automated, fully automated, because it has these robots. This one is actually called robots because it can be moved, yeah, along this platform to carry out sampling and also measurement of the OD. So some of the microbioreactors, it has in situ measurement of the OD. The one that I use, this one is actually, I use it during my PhD studies. It is semi-automated, whereby I still have to do the sampling and the addition of the media manually. So I have to insert the media one by one. But it's not that really tedious because I have these multi-channel pipettes. So I can pipette like 4 wells at the same time. Yeah? So, but those things have to be done manually. So that's why it is called semi-automated. The rest of the things like the shaking, the control, and the monitoring, that one can be automated. So that's why it's called semi-automated. But for this one, this is a fully automated system whereby everything is automated. It doesn't need the manual intervention, yeah, the human intervention. So what the humans will do is just to control everything on the computer, just to set the set point of the pH, the temperature, the time for the sampling, and everything will be done by this robots. And this is actually called robots because it will do the things inside of the human. Another example, this one I think I have showed to you in the last few slides. This is Micrometrics by Applicant Biotech. Applicant Biotech is the name of the company. Micrometrics is the name of the bioreactor. Yeah. So the characteristics is different from one brand to another. Yeah? But this one, it has, both of them have 24 wells with individual pH, dissolved oxygen and temperature control, and also the bioreactor volume is the same, which is 10 milliliter for each of the well. Yeah? So some of them have similar characteristics, but in terms of the design or maybe the operation is different from one brand or from one model to another. And this is another example of a more complex system, a more advanced system, which is called Amber 250, the name of the bioreactor. And the company that produced this system is Sartorius. Yeah? So this one, the design, you can see that the design is different. And this actually mimics the conventional vessel of stirred tank bioreactor. Yeah? But it is small in size. And bear in mind that the material of this is actually plastic. It's not glass or it's not stainless steel. So it is disposable and it is to be used for single use only. So it has the disposable feature, apart from the high throughput and also the micro scale feature. So this one is fully automated. You can see this robot sits here, right? So in practice, during the operation, this one will actually move along the platform over here to carry out the sampling and also the analysis and also the even the media loading. Yeah? It can do all the loading by itself without the human intervention. This is another example. This is called Desbox Mini Bioreactor, which is produced by Eppendorf, the name of the company. And this platform is different. It only has 4 bioreactors, right? And the size is different, the design is different. This one looks like very much like the conventional stirred tank bioreactor with the impeller inside. Yeah? And this is how the system, the whole system is connected to the computer. Yeah? And here is the summary of several studies that have used microbioreactors and mini bioreactors. And the applications is very wide. It's not just applicable to one type of organism. But you can see that here, like Micro24, this is actually my work. I use it for fermenting E. coli, producing transaminase, which is an enzyme. But other applications of Micro24, it also applicable for mammalian cell cultures, Chinese hamster ovary. This one has been reported by Chance et al. And Batts et al. In 2009 and 2014. You can see it's quite some time ago, actually. Apart from the CHO, it has also been used for S. cerevisiae, Saccharomyces cerevisiae, P. pasteurus, which are both yeast, and also E. coli. This one has been reported in 2007. And that one is Micro24. Other types of bioreactor are bioreactor system, which has been reported for E. coli and Hansenula polymorpha, reported by Kansi 2009. Other platforms, Micrometrics, has been used for fermenting Chinese hamster ovary, which is a mammalian cell culture, right? And also Amber system is also used for Chinese hamster ovary. So normally, most of the works revolves around the mammalian cell culture because those hosts are used for producing pharmaceutical or therapeutic proteins, which are expensive. So that's why the interest is actually more for the mammalian cell cultures. But still, the platforms are also applicable for microorganisms like the bacteria, yeast, and fungi. So the last slide for this subtopic is this diagram, which shows the comparison of features between standard shake flask, the conventional shake flask system, the high throughput control microbioreactor, which sits in the middle, and also the conventional bioreactor. So let's have a look at first the shake flask system, which is shown by the first circle over here. So the shake flask, it is the oldest technology. It doesn't have the environmental control. We cannot control the pH. the temperature can be controlled only when it is placed in the incubator, but that is ambient temperature, not the temperature inside the flask. So in general, it doesn't have the environmental control. So that is the downside of the shake flask system. But the advantages of the shake flask is that it requires low material compared to conventional bioreactor, which normally exists in 1 liter at least. Yeah. So in general, shake flasks require much less materials, lower materials, right? And it is easy to be handled. So you can clean the flask easily, and you can autoclave it easily without much problems. Yeah? So in general, it is easy to be handled. So that's the good side of the shake flask. Now let's move to the conventional bioreactors. The good side of the conventional bioreactors, it has the environmental control, unlike the shake flask. It has the pH control, it has the temperature or dissolved oxygen control, and even the mixing capacity is much better compared to the shake flask. But the downside of the conventional bioreactors are the low throughput, which means that it normally the conventional one exists only in like a single unit. Yeah, so that is low throughput, and because of the size is big, the size is perhaps like the lowest or the smallest size is like 1 liter, so it require high material, high amount of media. Yeah. So you cannot run a 100 milliliter fermentation in 1 liter fermenter. So it is not possible. So that's why it require more media, more amount of media compared to the shake flask. You need at least 800 milliliter of media to be carried out in 1 liter fermenter. So in general, it requires high amount of materials or media. And then it is labor intensive in terms of the handling and also the operation. It means that, it's very laborious to clean it, to sterilize, to prepare the media, to set up everything and so on. And if let's say you perform, like if you carry out fermentations in more than 1 unit of bioreactor, it might need manpower, more manpower to handle the operation. Okay, so in general, it is labor intensive. So based on these advantages and the drawbacks of shake flask and also conventional bioreactor, so that is why high throughput control microbioreactor is introduced. Yeah. So this high throughput control microbioreactor, it tries to integrate the advantages of both conventional platforms, shake flask and the conventional bioreactors. So it having the micro scale or mini scale feature, it doesn't require high amount of materials. So it means that it requires low materials. And then because of the small size, it is easy to be handled, right? So which is similar to shake flask. But it eliminates the drawbacks of the shake flask by having the environmental control, which is the feature of the conventional bioreactor. So even though it is small in size, but it has the environmental control for all of the wells in the platform, yeah, for each of the wells even. Yeah, so it has a pH control system, temperature, and also the dissolved oxygen control system. And additionally, it has the high throughput feature, means that it does not exist in only 1 unit, but it is exists in, let's say 6 or 12 or 24 or more than that. Yeah. And also there is an automation feature. Automation means that everything can be handled simultaneously at and standardized, yeah, right? So automation is also embedded into high throughput control microbioreactor. So that is how this platform can perhaps improve the fermentation performance. It can help to reduce the timeline for the process development, in the industry. So instead of, let's say, maybe 5 years of process development, if let's say we use the conventional platform, we can reduce the timeline to maybe 3 years, because we can carry out many fermentations at the same time in order to get the optimal parameters. So that is the development stage of any products. So in general, the high throughput control microbioreactor with the new concepts, for example, the micro or mini scale disposable technology, and also the high throughput technology, it can benefit the fermentation process. And that is how the modern fermentations are carried out in comparison to the traditional fermentation processes.